Dear student, welcome to our program, Knowledge Quest. The subject is biology, and I'm your teacher, Lydia Asiago. In our previous lesson, we have been learning about the process of digestion, and so far, we have discussed what happens during digestion, that is at the mouth. We moved on to the stomach, in the duodenum, but we still need to complete the whole process of digestion. Now, what did we say during the process of digestion? We need to find out if we can remember so far what we have discussed about digestion. And remember, we had an example of a person who ate a meal that consists of starch, proteins, and lipids. Now, remember we said the process of digestion starts in the mouth. What happens in the mouth? Remember, we are just reviewing what we've been learning so that we are able to connect to the next of the lesson. Now, we said that in the mouth, there are processes that takes place. Number one, we have the chemical breakdown of food, and still we have mechanical breakdown of food. Now, we said that the moment you introduce food into the mouth, number one, we stimulate the salivary glands that secrete saliva. We said that saliva is a mixture of substances which include mucus, water, salivary amylase as the enzyme, salivary amylase. And of course, we were able to give the function of each content in the mixture that is in saliva. So we said that mucus lubricates the food, water moistens the food, salivary amylase, we said that this one breaks down the starch in the food into maltose. Now, we went ahead and also said that in the mouth, we have the teeth, and the teeth does the mechanical breakdown of food, whereby we said there is a process that takes place, a mechanical process that we call mastication. Remember, we are reviewing what we have learned so far in the process of digestion. And then we said that the teeth breaks down the food substances that are large into small substances in order to increase the large surface area for the enzyme to function. And we said that once the food has been broken down, the only chemical process that takes place is starch being broken down to maltose. And then we also said that in the mouth we have the tongue, and the work of the tongue is to roll the food into bolus. And then we said that the boluses, they are moved at the back of the mouth, and then they move down the digestive system, that is down the esophagus, through the process we said peristalsis. Peristalsis. So briefly, this is what we discussed on the process of digestion in the mouth. Then we said that the moment the food gets into the stomach, which is the next part of the digestive system, we said that the presence of food in the stomach stimulates the stomach walls to secrete a hormone by the name gastrin. So we moved, this is the first point. The second point we discussed is in the stomach. And we said that in the stomach, the moment the food gets into the stomach, it stimulates the stomach walls to release a hormone by the name gastrin. And we said that gastrin in turn stimulates the gastric glands that secrete a digestive juice by the name gastric juice. Gastric juice. Then we said that gastric juice consists of, number 1, hydrochloric acid. Number 2, gastric juice consists of enzyme by the name pepsin. And we said that pepsin is produced in an active form by the name pepsinogen. We also have the enzyme renin. And so far we said that we also have mucus in the stomach. Now what did we say? We said that digestion in the stomach, the enzymes that break down food in the stomach, they require an acidic environment for them to function well. And that's why we say that we have hydrochloric acid. And we say that hydrochloric acid has 3 roles. The first role, we said that it provides acidic pH, provides the acidic pH for enzyme pepsin and renin. We also said that hydrochloric acid activates the inactive pepsinogen to pepsin. And the third role of hydrochloric acid, we said it kills some of the bacteria that might be present in the food. We went ahead and discussed the role of pepsin. We said that pepsin breaks down the proteins into peptides. So we said this one breaks down the proteins into peptides. So far, we are seeing that in the stomach, there is digestion of proteins, and we say that in the mouth, there was digestion of starch. Now, what happens? We also said that we have mucus, and the work of mucus is to coat the lining of the stomach, and in a way, it protects it from digestion of by the protein digestive enzymes. The protein digestive enzymes might digest the wall of the stomach, and that is why it has to be protected by the mucus. And of course, we also know that mucus lubricates the food, so food moves easily in the digestive system. Something we have to remember is that we say that for food to move down the digestive system, that is from the mouth through the esophagus to the stomach all the way to the duodenum, there is a process that takes place, a process we call peristalsis. So we should have that in mind. Now, then we said that because there is a lot of mixing of these food substances together with the digestive enzyme, we said that the end product or in the stomach we are forming is a acidic chyme. And this acidic chyme moves all the way to the duodenum, which is the first part of the small intestine. So number 3, we are moving to the duodenum. Now, duodenum is the first part of the small intestine. The small intestine is divided into 2 parts. We have the duodenum, and the second part, which is the largest, we have the ileum. Now, what did we discuss? We said that in the duodenum, the moment the acidic chyme gets into the duodenum, it stimulates the duodenal walls to secrete 2 hormones. The first hormone we said and discussed is secretin. We also discussed about another hormone by the name cholecystokinin. Now, so far we said that secretin stimulates the liver. We said this one stimulates the liver. And what does the liver do? The liver releases the bile. And we also discussed what is the role of bile in digestion. We said that bile has two functions. Number one, it neutralizes the acidic chyme from the stomach. Remember, in the stomach it is acidic because of the hydrochloric acid, but in the mouth it is alkaline because saliva is alkaline in nature, so it is basic. But you realize that the enzymes that function well in the duodenum, they need a pH that is alkaline. And that is why we are saying that bile neutralizes the acidic chyme. And number two, we said that bile does emulsification. Emulsification. So here we said it does the role of neutralization of acidic chyme. And it also does emulsification. This is just what we discussed. We are just going through it so that we are able to connect to today's lesson. Now, what else did we discuss? We said that there is another hormone called cystokinin. And the work of this hormone, it stimulates the pancreas. Stimulates the pancreas. So we have said that in the duodenum it is alkaline. It is alkaline. Now, I think that is where we stopped. So we need to continue from there. What happens after the liver has released the bile? The bile, which number one does the neutralization. So we want to assume that our chyme is now alkaline. We also have the bile that has done emulsification. Now, another part or another process that takes place, you realize that the second hormone, which is called cystokinin, it is the one that stimulates the pancreas. It stimulates the pancreas to release pancreatic juice. Pancreatic juice. Now, pancreatic juice is a mixture of substances. There are different components in the pancreatic juice. Number one, in the pancreatic juice, we have pancreatic amylase. Pancreatic amylase. Number two, we have trypsin. Trypsin is an enzyme. And number three, we also have sodium hydrogen carbonate. Sodium hydrogen carbonate. Sodium hydrogen carbonate. So those are the components. Those are the components of the pancreatic juice. We also have another one, that is the pancreatic lipase. I have not mentioned about the pancreatic lipase. Remember, we are discussing the contents of pancreatic juice. I think we are going to list them again so that we see the function of each of the components of the pancreatic juice. So we are saying that we have pancreatic juice, which consists of, number one, pancreatic amylase, number two, trypsin, number three, pancreatic lipase, and number four, there is another salt by the name sodium hydrogen carbonate. Now we want to see what is the role of each of these components of the pancreatic juice. Now remember, starch was first digested in the mouth by the enzyme salivary amylase. But when we moved to the stomach, the digestion of starch stopped. The salivary amylase could not continue digesting the starch. Why? Remember we said salivary amylase works best in a medium that is alkaline. But you realize that in the stomach it is acidic. And that is why the functioning of salivary amylase will stop in the stomach. So in a way we are saying there is no digestion of starch in the stomach. So what happens? The starch is digested in the mouth. Then the next place we are going to meet the digestion of starch is in the duodenum, whereby we have the pancreatic juice, which consists of pancreatic amylase. So what will pancreatic amylase do? Remember, some of the starch in the mouth was broken down to maltose, but not all the starch. So what happens is that the pancreatic amylase will break down the remaining starch into maltose. So we are saying the remaining starch. Why are we saying remaining starch? It's because already we had digested some of the starch in the mouth. The remaining starch is broken down to maltose. Meaning at this point, we expect at least all the starch to be broken down to maltose. Then trypsin, remember in the stomach, that is where we first digested or we broke down the proteins. So the proteins were broken down to peptides, but not all the proteins were broken down. So trypsin will also break down the remaining proteins into peptides. So you expect at the duodenum, all the proteins have been broken down to peptides. In the duodenum, all the starch has been broken down to maltose. And then the first point where we are seeing the digestion of lipids is in the duodenum. This is the first enzyme that we are coming in contact that is going to break down the lipids. So the lipase will break down the lipids into fatty acids and glycerol. Pancreatic lipase will break down the lipids. So the first point we are seeing the digestion of lipids is in the duodenum. The lipids will be broken down by enzyme pancreatic lipase into the fatty acids and glycerol. So it means that not all of the lipids will be broken down. That is maybe some. Now, what is the role of sodium hydrogen carbonate? Remember, this is a base. And we know that anywhere we have a reaction between a base and an acid, that is a process we call neutralization. So in a way, the work of sodium hydrogen carbonate will still do the part of neutralizing the acidic chime that got into the duodenum. So far, that is the brief digestion of the brief process of digestion in the duodenum. We just want to break for a short commercial break, then we will be back. Just stay tuned. Dear student, welcome back to our program Knowledge Quest. Our subject is biology. And before we went for a break, we were discussing the process of digestion in the duodenum, whereby we said that in the duodenum, we have the pancreatic juice, which consists of the 4 components: pancreatic amylase that breaks down the remaining starch. And I said that remaining starch because some of the starch had been broken down in the mouth. So the remaining starch is broken down to maltose. The remaining proteins, remember, were first broken down in the stomach. Those that have not been broken, they are broken down by the enzyme trypsin to the peptides. That is, proteins are broken down to peptides by the enzyme trypsin. Then we say that the first point we are seeing the digestion of the lipids is in the duodenum, whereby we said that we have pancreatic lipase that breaks down the lipids into fatty acids and glycerol. And then we say that because the acidic chyme that gets into the duodenum, we need to neutralize it. That's why we say that we also have another component of sodium hydrogen carbonate, which is alkaline in nature. And this one will neutralize the acidic chyme. So at the end of the day, we say that for the enzymes to function well in the duodenum, they need an alkaline environment. And that is why we are saying that we have to neutralize the acidic chyme. Who did that first? We had the bile, that is the bile salt. And number 2, we are seeing sodium hydrogen carbonate doing the same role of neutralization. Now, after that, we expect there is a lot of churning, mixing of the food substances together with the digestive enzyme. So we expect this acidic chyme, which has been neutralized now, it is alkaline. We expect it to move to the next part of the digestive system, which is the ileum. Remember, we said that from the stomach, we have the small intestine. And we say that the small intestine is divided into 2 parts. The first part is the duodenum, and the second part is the ileum. So you want to see what happens in the ileum. That is digestion in the ileum. Now, once the chyme, now we are calling it the chyme, gets into the ileum, it will stimulate the ileum walls to secrete an enzyme, a digestive juice by the name intestinal juice. It is going to secrete an enzyme by the name intestinal, we say intestinal juice. Remember, I said that the arrival of that chyme in the ileum will stimulate the walls of the ileum to secrete a digestive juice by the name intestinal juice. Now, what is the function of the intestinal juice? First, let's see the components of the intestinal juice. Number 1, we have maltase. Number 2, we have sucrase. Number 3, we have lipase. Number 4, remember I'm listing the components of intestinal juice. Now, who releases the intestinal juice? It is the ileum walls. And why does the ileum walls release the intestinal juice? The presence of that chyme in the ileum will stimulate the walls of the ileum to release a digestive juice by the name intestinal juice. Now, the intestinal juice consists of maltase, that is an enzyme, sucrase, lipase, peptidase, and number 5, there is also an enzyme, lactase. Now, components of the intestinal juice. Maybe to think back, which components of food that entered the ileum? Which components of food that entered the ileum? Number 1, we had maltose. Where did we get maltose from? The starch was broken down to maltose. Number 2, we have peptides. Peptides. Where do we get the peptides from? Sorry, this is peptides. Where do we get the peptides from? From the breakdown of proteins. Let's make it clear. So we have the peptides. Peptides from the breakdown of proteins. And then number 3, whatever that is moving to the from the duodenum to the ileum, we also have the lipids. The lipids that were not broken down. Although we still have the fatty acids and glycerol. But we don't expect them to be digested. So these 3 are from the duodenum. And that is why we have enzyme maltase. Now the work of enzyme maltase is to break down the maltose. So in this case, it will break down the maltose into glucose. Enzyme maltase will break down the maltose into glucose. The enzyme sucrase. Now you may ask, why are we talking about sucrase? Maybe someone else ate food that is rich in sucrose. So we can have sucrose in the diet. We can have lactose in the diet. What will happen? The sucrase will break down the sucrose into glucose and fructose. Sucrase will break down the sucrose. That is, if we have some of the sucrose in the meal, because these are the components of the digestive juice, which is the intestinal juice. So we have to mention all the components in the digestive juice, so that at least we give the function of each. Then we have lipase. Lipase will break down the lipids. The lipids that were not broken down in the duodenum, because the lipids were first broken down in the duodenum. So some of them that were not broken down will be broken down by enzyme lipase into fatty acids. So this one will be broken down to fatty acids and glycerol. Now, peptidase. What is the role of peptidase? Peptidase will break down the peptides. Remember, the peptides, we got them from the breakdown of proteins. So we'll break them down into amino acids. So peptidase will break down the peptides into amino acids. Then we have lactose. Lactose breaks down any lactose that might be present in the diet into glucose and galactose. So basically, what are we saying? We are saying that ileum has 2 functions. Number 1, it is the one that completes the whole process of digestion. Remember, when we started this lesson, we were asking ourselves, what is digestion and why do we need digestion? We said that we want to break down the complex food substances into small molecules that are soluble and absorbable, so that we can absorb them into the bloodstream, whereby they are transported to various parts of the body, either for use or for storage. So basically, you realize that the complex food that we had, that we were breaking down, we've already broken them down to the simple molecules. Which are these simple molecules? We started with starch. Starch has been broken down to glucose. as the simple molecule. Now you realize that starch is an example of a polysaccharide. Polysaccharides are always broken down to disaccharides. In that case, we have the maltose, which is an example of a disaccharide. Then the disaccharides are broken down to monosaccharides. Remember, monosaccharides, these are substances that are very simple, they are soluble, they are sweet like glucose, and they're easily transported in the body system. That's why we are breaking down the starch to maltose and then to glucose. Then we are saying that the lipids are complex molecules, which we've also broken them down to simple and absorbable molecules, which are the fatty acids and glycerol. And then the fats, all the proteins have also been broken down to amino acid. So basically we are saying that the first function of the ileum, which we are able to see here, is that it completes the process of digestion. Now basically what are we saying? What is this that we've broken down? Let's trace it. What were we digesting? We were digesting starch, proteins, and lipids. You are digesting starch, proteins, and lipids. So we are saying that the starch that we had, starch which is an example of a carbohydrate, and we say that carbohydrates are complex, they are grouped, they are classes of carbohydrates. We have polysaccharides, we have disaccharides, and then we have monosaccharides. So we picked an example of starch as a polysaccharide. We picked an example of starch as a polysaccharide. And we are able to say that starch has been broken down to maltose. Then finally, maltose has been broken down to glucose. So it means that the glucose that we are going to have now in the ileum, glucose, is number one, simple, it is soluble, and it is absorbable. So it can be absorbed into the bloodstream. That is now the whole summary of what? Starch, which is a complex carbohydrate, being broken down to a simple carbohydrate, which is glucose. Then we also saying that the proteins which we had in the meal, the proteins are broken down into peptides. Proteins are broken down to peptides. Then the peptides are broken down to amino acids. So what does that mean? It means that the complex proteins, which are insoluble, which cannot be absorbed into the bloodstream directly, have been broken down to simple, soluble, and absorbable product, which is the amino acids. Then we also saw that in the whole process of digestion, we are digesting the lipids. If we trace the breakdown of lipids, we say that lipids were broken down to fatty acids, and glycerol. Meaning, the fatty acids and glycerol, they are simple and they can be absorbed into the bloodstream. So basically, what are the end products of digestion? The end product of digesting the starch, or let me say, basically the carbohydrates is what? Is glucose. Why am I saying glucose? Because somewhere else we saw that we were breaking down what? Sucrose into glucose and fructose. So at the end of the day, we also having glucose. We also saw that we are breaking down the lactose into glucose and galactose. So at the end of the day, the carbohydrates that we expect to have, the carbohydrates that we expect to have, that is after digestion, carbohydrates, which are now the monosaccharides. We expect to have glucose, galactose, fructose. So in a way, the complex carbohydrates, all the polysaccharides are broken down to monosaccharides, and the products of monosaccharides we have is the glucose, galactose, and fructose. But you are going to see that the moment the fructose and the galactose are absorbed into the bloodstream, at the end of the day, they are going to be converted to glucose. So at the end of the day, the carbohydrates are broken down to glucose. So basically, this is what we've been digesting. So the process of digestion starts in the mouth and ends in the ileum. And then, of course, we ask ourselves, after digestion ends, what happens? It is the process of absorption. But before we go to the process of absorption, it is good we have some points to note. We need to note this, that enzymes, if you remember very well, enzymes, the digestive enzymes, or enzymes generally, enzymes are protein in nature. And because they are protein in nature, there are properties that they have and there are factors that affect their functioning. And in the whole process of digestion, we picked on one property or one factor that affects the functioning of enzymes, whereby we talked about what? The pH. pH is a key factor that will affect how the enzymes will function well. And that's where we have to be very keen. There are those enzymes that work best in alkaline. Now, in the digestive process, there are those enzymes that work best in acidic environment. Like now we said that in the mouth, it was alkaline. And which enzyme did we see in the mouth that works best in alkaline? We saw just one enzyme, salivary amylase. Salivary amylase. Salivary amylase. So these are points as a student that you need to take care of, because you can be asked, name the enzymes that work best in acidic environment, that is in the digestive system. Name the enzyme that work best in alkaline environment. That's why we are making this summary that in the mouth it is alkaline, and the enzyme that can function best is the salivary amylase, because the pH is okay for that enzyme. Then we say that in the stomach, it is acidic. And the only enzymes that we realized that were found in the stomach, they were two that work in the acidic environment. We talked about pepsin. And number two, we talked about renin. Those are enzymes that are working best in acidic, that is in acidic pH, acidic pH. Then we move to the duodenum. We said that those enzymes that work best in the duodenum, they need an alkaline environment, a pH that is basic. And which are these enzymes? Number one, we talked about trypsin. And if you are very keen, trypsin is an enzyme that digests proteins. Pepsin is an enzyme that digests proteins. But you see the two enzymes, they are working totally at a different pH. Pepsin works best in acidic, yet it breaks down proteins to peptides. Trypsin also works best in alkaline, but it also does the same role, breaking down the proteins into peptides. That's why we have to be very keen that enzymes are very sensitive to change in pH. And that is why you realize that salivary amylase could not function in the stomach, because in the stomach it was acidic. I hope that point is clear. Then we also have another enzyme that is working in alkaline environment, that is in the duodenum. We have another enzyme, we say pancreatic lipase. Number three, we have pancreatic amylase. These are three enzymes that we find in the duodenum that work best in alkaline. And if you are very keen, the enzyme that break down starch, pancreatic amylase, it is working in alkaline. The same case, salivary amylase, it is working best in alkaline. So basically, the issue of pH and the functioning of the enzyme, you have to be keen, is that one. Then, of course, we expect the enzymes in the ileum. Enzymes in the ileum, they're also in alkaline environment, because from the duodenum, remember we did the neutralization. So whatever that we have is neutral, it's not acidic. And that is why we are saying that in the ileum, those enzymes are not working in acidic environment. They are working best in an alkaline environment. Now, that is the issue of pH and digestion. Now, another summary that we need to take note in the process of digestion, another summary as a student that you need to take note in this process of digestion is now to summarize the whole process of digestion in just a simple table. For example, we can say which part of the digestive system we have, the part of digestive system. So we want to make a simple table that will help us do the whole summary of digestion. So we have the gland that is responsible, which gland or glands that are responsible. Then we can also have the issue of what does it secrete. So the enzyme, the secretion, what is it? Let me say the gland and the secretion. And what does it secrete? Secretion. Then the enzymes that are present. Enzymes that are present. And then the food substrate that is being broken down. The food substrate digested. Then we have the end product. So this is just a final table to summarize the whole process of digestion. So we started from the mouth. We started from the mouth. So we say that in the mouth we have the gland. Which gland is that? Salivary gland. What does it secrete? It secretes salivary. It secretes saliva as a juice. That is the secretion that it is released. So salivary glands, they secrete saliva. Which enzyme is present? We have salivary amylase. Salivary amylase. What does it do? It breaks down starch. At the end of that process, what is happening? We are producing maltose. Then we move to the stomach. What happens in the stomach? We say that in the stomach, there is a gland. Which gland is that? Gastric gland. Gastric gland. What does it release? Gastric juice. Gastric juice. This is just a summary for those who have not been with us. If you have just joined us, if you have this summary, I think you can digest that information. Gastric juice. Which enzymes are present? We said we have pepsin. What does pepsin do? Breaks down the proteins. And the end product is peptides. Then we also have renin. What does it do? It breaks down, or it curdles the, we say that it curdles the protein milk. Protein milk. Or milk protein. Let me call it the milk protein. we have the milk protein into, of course we say that the moment the renin curdles, it converts the caseinogen into casein. Then those are the enzymes that are working in the stomach. Then we also have, from the stomach, we have the duodenum. Then in the duodenum, we say that we have, which gland releases the juice? It's the pancreas. The pancreas, what does it do? It releases pancreatic juice. Pancreatic juice. Then the pancreatic juice, we say that it consists of the pancreatic lipase, pancreatic amylase, and lastly we said we have trypsin. This one breaks down, pancreatic lipase breaks down the lipids. What are the end products at that process? We have fatty acids and glycerol. Fatty acids and glycerol. Then we said pancreatic amylase will break down the maltose, the starch that was not broken down in the mouth, that is the starch, into maltose. Then trypsin will break down the remaining proteins into peptides. And lastly, whereby we say that the process of digestion ends in the ileum. And in the ileum, we say that we have the intestinal juice. So the juice that we have here is intestinal juice. Intestinal juice. And then intestinal juice, we say that we have maltase, we have sucrase, we have lipase, we have peptidase, and of course we said that the end product of the maltase will break down the maltose into glucose. The sucrase will be broken down to glucose. That is, will break down the sucrose into glucose. And then the lipase will break down the lipids, the remaining lipids, into fatty acids and glycerol. And lastly, peptidase will break down the peptides into amino acids. And that marks the end of the whole process of digestion. So, my dear student, that is the summary of what we've been discussing about digestion. And maybe in our next lesson, we are going to discuss now what happens to the end products of digestion. I'm saying in our next lesson, you see now the end products of digestion, we have glucose, we have fatty acids, we have amino acids. So we are going to discuss and see what happens to the end products of digestion. And that's why I want to ask you as a student, always stay tuned to our program, Knowledge Quest, and the subject is biology. I've been your teacher, Lydia Siago. See you in the next lesson.